the 2 decades that he's been working on this problem, which began as a basic science project to understand how unfolded proteins are dealt with, are managed and degraded in eukaryotic cells, culminating in understanding the mechanism of action of an important anti-cancer drug, which is now also the target for the compound here. Kyprolis, I believe, is the name. So, Michel, without further ado, it's an honor to welcome you to the stage for this keynote. Thank you very much for agreeing to speak.
Good morning, and thank you for your kind introduction. Thank you for being here, and it's my pleasure, and I appreciate to hopefully entertain you for the next 50 minutes on 20 years of proteasome research, at least what my group and myself participated. I decided to focus in particular of the crystallographic part, what we've done, and definitely it's a challenge. So the topic will start, as we see it here, about the life cycle of proteins, who everybody knows here, starting from individual amino acids and ending up at proteins, folded proteins, at specific locations. But once the function is fulfilled, or the proteins have no further need, degradation occurs. And that is the topic I want to talk about, protein degradation. It is a way that exists several proteases, but in eukaryotes, the major key player of the non-lysosomal protein degradation pathway is the proteasome. And in the talk today, I will only focus on the core particle of the proteasome, the 20S proteasome. The system is much more complex, but as you will see, the 20S proteasome, so only the proteolytic chamber is still very complex, at least for us to understand. Now, the proteasome, I will go in a little bit of more detail, but you see it here in eukaryotes. That's the size of the proteasome, how it is composed, and it is involved in a lot of intracellular irreversible processes. So it is a controlling machine, and therefore it has a lot of potential applications, but you can also cause a lot of damage by manipulating it, and that is also exploited by a lot of natural products, a lot of pathogens who try to modify the activity of the proteasome. We see it here shortly in cell differentiation. We're having a heart cell or a nerve cell. So thus it is involved in the degradation of transcription factors, because the 2 cells I've mentioned, they share the same genomic identity, but they are not convertible in each other. In cell cycle control, you see it on the left top side. So that means that the proper cell proliferation takes place. You need protein synthesis, but also protein degradation. So some proteins have to be removed from the cell so that the cell can continue, and that is exploited by cancer therapy, for example, by inhibiting the proteasome to cause a cell arrest. We're having down the antigen presentation. That means we're having various subtypes of proteasomes. I will come to this talk, how you produce antigens which are presented on the cells. And on the right side, the stress response. So that explains that it is an interplay between molecular chaperones, unfoldases, when you're having aggregates, and protein breakdown. Now, showing that the proteasome is involved, I want to explain you about the milestones in structural biology of the 20S proteasome. And you see it on the left side. It started where the proteasome was not known what the function is about it by a single author paper of Harris, who goes to erythrocyte ghosts and who find large particle insights. That were the first electron micrographs on the proteasome. And then it was in 1988 where the electron microscopy improved by Baumeister et al. Paper. and that was the first size how the molecule looks like. It was at the time when the proteasome function was also addressed. It was in 78 about, it was the groups of Goldberg and of Rechsteiner in particular. They could show for the first time that it's a protease, and Goldberg, it was also in 1988, introduced the name proteasome to it as the antagonist of the ribosome. The famous paper came out in 1995 by Löwe et al. So that was the first crystal structure of a proteasome from Thermoplasma acidophyllum. It was a time when I started my diploma thesis in Huber's department. So I just arrived in time, and I got as a project the eukaryotic proteasome from yeast, and that was published in 97. And then you see it here between 2002 and 2015, there have been published proteasomes from mammals. Now, being in history, I want to show you about a little bit more about the proteasome, which you see it on the next slide. So the proteasome is having a typical architecture. Just focus here on the Thermoplasma proteasome. It is composed of 4 stacked rings, each ring having 7 subunits, and there exists only 1 alpha type and 1 beta type subunits in this more simple proteasome. So the architecture, you see it on the right side, the picture is quite complicated, but you can see that the overall architecture is always the same on 20S proteasomes. And their proteolysis, so the active centers are located at the beta subunits, and they are the 2 inner parts of the ring where now the pointer is showing. And in case of the eukaryotes, so the more simple eukaryotes are yeast or fungi, for example. So in this case, single cell types. They harbor 1 type of proteasome that is shown here. And as active sites, they're having 7 different alpha type and 7 different beta type subunits. And it is the beta sub, which having 7 distinct subunits, so the molecule having a C2 symmetry, so we change from D7 to C2. And in this talk, I will predominantly focus on subunit beta 5 of this one. And later on in this talk, I will discuss about the mammalian proteasomes, so they're having the same set, except that the active sites get replaced. It needs a de novo synthesis, and now the 3 active sites, beta 1, 2, and 5, got replaced by immunosubunits. And in case of beta 5, there exists a thymoproteasome. So you're having 3 distinct active sites, beta 5C, beta 5I, beta 5T, in respect to this active subunit, so a diverged evolution. Now the proteasome fold is old. You see it on the left side. It's an N-terminal nuclease fold as first introduced by Brannigan in the 1995. But it is old. It is old because Wallace and Burns introduced by bioinformatics studies that already cyanobacteria, which are more than 3.5 billion years old, have such a fold and produce such proteins. So we are talking about an old protein, and that reminds me to the introduction slide. We are talking about the life cycle of proteins, and therefore, otherwise we would be not sitting here. Proteasomes are almost as old as ribosomes because you build up and you construct the system to get your natural resources, individual amino acids. Now, the proteasome is a very dangerous particle, and it's one of the most unselective enzymes in the cell because it can cut complexity down to size. I'm referring to this because the 97 paper was accompanied by News and Views from Jones and Stewart, and they produced the title Cutting Complexity Down to Size. And therefore, since it can cut about more than 90% of proteins, non-lysosomal in the cell, it needs to be a strict regulation. And the question is about how do cells build up such a complex molecular machinery or degradation machinery. Now, this is a simple way which we can see here about how proteasomes assemble. And we're having individual alpha subunits and beta subunits, and as mentioned in the more simple case, you also were working on Archeglobus fulgidus proteasome. It's another archebacterial proteasome. We succeed in a structure, and this structure looks like it is shown here. We're having 7 alpha subunits forming spontaneously a ring. Now the beta subunits, as you see it on the slide, having this red mark, they are not produced in their active forms. They are produced in when one works with protease as cymogens, so they're having a precursor sequence, and they cannot assemble. They cannot assemble because they lack, not end show, that would be bad, because they lack this insertion loop. So it's a contact surface. And they need the alpha ring to form and to assemble it in a way like this. And that is shown here. So we make a mutation, a 3-nivalenol mutation in Archeglobus fulgitus. So we only need 2 kinds of proteasomes, and therefore we can also look and trap assembly intermediates. It's a story about when I'm talking here about 2000, in these years, 2000, 2004. And we see that the 2 half proteasomes already sit to each other, but there is a gap, and it cannot succeed in the assembly because the precursors cannot be removed. So I'm coming a little bit what the active site of the proteasome is about. It's a 3-in-1 protease. Now, the next slide then is, can we analyze how these steps of activation occurs? And we were working with yeast. We placed only 1 active site, so these yeast cells are viable, and we put in a 3-nivalenol mutation in subunit beta 1. Yeast genetics has been performed by Heinemeyer in the group of Wolf at that time, and we combined, and we see on the right side of the image that now the 1 precursor cannot be removed. You're seeing here the point of the alanine 1. It's in principle a threonine, and we can suggest mechanisms of autolysis, and that is defined electron density. So we understand that this is a very late event in the assembly pathway, and that shows up that we remove, when you see it on the left side, the beta subunit, which is shown here, remove this red cymogen pre-segment structure, but we're having a new gate. We determined it as a latent proteasome because, as you will see on the next slide, the proteasome is sealed. So we're having, and that is the title of what you can see, a coupled regulation of proteasome assembly and inhibition. And now we stopped and we make a process that the first auto-inhibition is removed, but we're having a sequestration of the active sites, as you can see it here. We named it latent proteasome because the structure in 97 clearly revealed that the active sites, so they are sequestered, similar as in the thermoplasma proteasome, in a central hydrolytic chamber, that you have sealed entrances. This is a half-cut view of the proteasome, which you show on the left side. And so we're having a gating principle responsible for it, as it is shown on the next slide. So we combined with Finley and we made point mutagenesis, and you see it here by only introducing a point mutant, aspartate 9 to an asparagine. So only replacing an oxygen to a nitrogen, you're seeing an open up of the whole plaque formation. So that shows that these residues are chemically there, but structurally distorted. And when you compare this with the 26S proteasome that is shown here, this proteasome has the same activity of the mutant one. It was viable in yeast compared to the natured or to the wild-type proteasome. So we learned at this time that rate-limiting step in proteolysis is accessibility because the active site didn't change. It was a back-to-back publication with the group of Hill, who could show it by analyzing regulators. It was fascinating, at least. Now we learned how proteasomes are set up, at least from a simple point of view in the cell, by a structural analysis. Now let's talk about proteolysis. This picture here shows the 97 work about the molecular resolution. And I want to slightly take you through it, how it's working. We're having a cis-al peptide bond, and this cis-al peptide bond, we're having a catalytic tride, which is not found in a lot of, or almost none other proteases, because it's a catalytic tride of aspartate 17, lysine 33, and threonine 1. This acts as a Bronsted base. You're having an alcoholate, and you attack with the alcoholate the carbonyl carbon atom, which is then finally trapped in an oxyanion hole. Now the next step is that however, proteolysis is not that simple because you're still having of the amine group a very bad leaving group, and that means we need a Bronsted acid. So to make out of the bad leaving group a good leaving group, and that means that we're having another catalytic triade, as it is shown here, which activates the N-terminus. So we're having a 3-in-1, which is the active site. And that means that since this is now protonated, that is our suggestion, we get out of here a good leaving group, and it is not this bond which is cleaved, which would be, when you don't protonate the amine group, the more reliable leaving group. It is the C-terminal fragment with the generation of a new N-terminus. You produce an acyl enzyme, as it is shown here, but now the 3-in-1 N-terminus is unhappy. It wants to get protonated back. And therefore it takes a water molecule, and this water molecule attacks the carbonyl-carbon, and finally, now you don't have here anything to protonate, and it is this group is getting cleaved. So what I want to show while I go in detail in this mechanism is the following. And therefore I make 2 citations. This is work from '97. This is work from 2016. Our latest suggestion of the mechanism. So suggestion of mechanism is challenging. And at least for us. Maybe you can also argue you are not that productive. But at least for us, to visualize in nowadays is more simple. But to put intellectual property inside so it might be reasonable is much more challenging. And as a chemist, this is causing also creativity and fun. Now, we determined with the mechanism a lot of inhibitors, and they are shown here. A lot of different classes. It's even only a small set. And I don't want to talk with all of them about you, but about 2 of you, the 2 of them. What I want to talk about are beta-lactons. I decided to talk about these inhibitors because beta-lactons were the first natural proteasome inhibitors found in '95 by Fenegal et al. And it was the first proteasome structure with an eukaryote and a natural proteasome inhibitor. What I want to show you, why I decided to use the mechanism might be simple. It's called omorolite or clastolactocystin beta-lacton. The reaction is quite simple because you attack with a 3-in-1 or gamma the carbonyl-carbon of the lactone ring, and so therefore you will succeed in this bond. But the surprise is that when you look of substrate cleavage, that you also produce this acyl enzyme intermediate. So the question is, why is the former an inhibitor, whereas the other one is an intermediate of protein breakdown? And that again shows, when you look at the structure here, that the, sorry for SLA, it should be omorolite, it is a nickname of it. It should be, in this way, a well-defined electron density. But it is bound, it inhibits. And the reason why it inhibits is because when you structurally superimpose it with the wild-type structure, that you get a displacement of the water molecule I've mentioned of less than 1 angstrom. And that explains, again, chemistry. So to cleave a peptide bond or to cleave an ester bond, you need a perfect Burgit-Donitz trajectory. And when you displace this trajectory, so you don't modify your protein, but the nucleophilic water molecule, this proteolysis cannot take place anymore. And again, I want to show you is, this is the structure from '97, and we took almost 10 years to get this understanding. So chemistry is complicated in interpreting and in suggesting mechanism. Now, the other thing is there exists from Salinospora tropica a compound that is shown here. It's a lactone, and this having this ethyl chloride group. I want to show this example because these are our electron density, what we get. They're nice to see. And you have to place it in. And when you place it in, you will not match the ethyl chloride group because you don't see it. It looks like this. You form a tetrahydrofuran ring. And that also explains about the mechanisms, what you see it here, that you're having a tetrahydrofuran ring formation. Now it's again protonated, of course, in this case, because it further continues, and that is termed radiocell activity. So it can be ongoing chemistry by natural product compound bonding, and this is an irreversible inhibitor because the tetrahydrofuran ring will never be open again. I want to choose another topic, and these are the second one of this list, the epoxy ketones. I choose the epoxy ketones Because it is an interesting chemistry. So it is in a way that it is a natural product, and that is, as you introduced, Stephen, already marketed. So that was the reason why I chosen it. So the system is looking on the top left. It is looking like that you're having an epoxy ketone compound. And that means the first step is quite clear. You attack with the carbonyl, with the 3-1 the carbonyl carbon atom. But what's happened next? What's happened next is no, that was it not. That should not happen next. So what's happened next is that should work, that this is the electron density. This is the experimental electron density, what you see, and you have to place it in, what's happening. And you see, this is a bulk electron density at the 3-in-1. Now, when you look at the compound, you see, now with the current mechanism, that you're getting a protonation of the epoxide, you're getting a carbocation, and we suggest a morpholine ring formation. And that nicely fits in the electron density. Now, I'm showing this. This was a work from 2000. And I'm showing this because what also can happen when we look again in the electron density, that there can also be a protonation of the epoxide, but a 7-membered ring formation. Kinetically may be advanced, but thermodynamically, of course, not compatible to a 6-membered ring system from chemistry. I showed this up because the 7-membered ring system also fits. And this was not our work, this was work in 2016. 16 years later, and they suggest it's not a 6-membered ring, so a 7-membered ring. The problem is that I would argue that there exist both rings. It's a classical iron theory that you have in kinetics and thermodynamics. And that is also that crystallography can be helpful, but you need to think about how to go on. And when one took now the morpholine ring or the ox-azepine ring system, in both cases, and that is the reason, so this is not that important for pharma industry, maybe for patenting, but not interesting because we cause a secondary R-, as you see it here. And either by a 6- or 7-membered ring. So it's an irreversible inhibitor, and it needs the 3-in-1 N-terminus, thus it is selective. And that shows it up. We're having a proteasome, as introduced by Stephen, which is a dirty drug. And we're having an inhibitor, which is FDA approved in 2012, which is a more proteasome-specific inhibitor. Identification of natural proteasome inhibitors. I was talking about 2 of them. How can we identify proteasome inhibitors? We can go and having a variety, in a huge variety of natural sources. And that is also the reason what I've mentioned. Proteasome is old, so there exist plenitudes of natural compounds inhibiting the proteasome. This is palaramine, one example on the left side down. Now one can go for simple testings, optical tests, but I want to show you that these high throughput tests, they are also demanding. Why I want to show you this? I want to show you how to identify a proteasome inhibitor or other inhibitors, and you can take what was very powerful in the mid of the last century, you can take isotope labeling. You take a peptide substrate, which can be cleaved by the proteasome, and you label it, as it is shown here, by C13. And once it gets cleaved by the proteasome, that means you either can even quantify and follow up the kinetics, similar as an optic assay, about proteolysis. And that, of course, when an inhibitor is inside, you will not see. Now I'm showing this. This is a Wikipedia image of Photorhabdus luminescens. It's a bacteria living in the gut of a worm, Heptaraptitis, and this worm is going through the soil and places its eggs in a larvae. And once it places its eggs in larvae, the larvae dies. It's a perfect biological symbiosis because the newborn worms feed on the larvae, they also feed the Photorhabdus luminescens, and then that is a biological system that the Photorhabdus luminescens supports evolution of the worm, and the worm supports survival of the bacteria. And this organism produces proteasome inhibitors. And when you're having crude extracts, when you grow them, because you want to know how, when these pathogens are produced, the proteasome inhibitors are produced, and how they look like. And when you're having such extracts or such crude extracts, you cannot use optical assays, but you can use the animal assay. You find that this one contains it, so you know what to analyze. You're making a reversed phase. You analyze that there are two proteasome inhibitors, which are split by a methyl group. You don't see it here, sorry, the image is too small, but it doesn't matter. And you then finally can analyze how this compound looks like. So you know when it is produced, you analyze that it is inside, and you visualize it. You see that is the different methyl group as you can see it here. Now, then, of course, you can go for structure and so on and to analyze the mode of action. The third thing, because we are here in crystallography, is this compound. So situation change, EM needs to go there. It's still taking time, so the success is great, what they did in the last 5 years. We can purify and crystallize thousands of yeast proteasome crystals within 24 hours, and a soak takes 12 hours. A data set at the SL synchrotron, which is greatly appreciated here, which is our home beamline in the meantime. You can collect a data set, let's assume within 2 to 3 minutes, including processing, that is very fast. You make a rigid body minimization with 2 cycles only, and then you can calculate an electron density and look for blobs. So that means, let's assume that this takes then not 10 minutes, but 15 minutes anyway. You immediately can use it when you're having some hits for screening. And that shows the next slide, because we're having natural products binding far off of the active site threonine, performing even here with the bromine halogen bonding. So you can, with sigma hole interaction, so you can see this, we will have a resnati. So we can see that we're getting a beauty of compounds, kendomycin binding far off of threonine, but also natural products. And you see you're getting a huge beauty to visualize following the system, and you're fast. Because of saving time, I don't want to go in the fragment evolution approach you've heard about from Professor Blundell, but I want to show you also about the beauty of crystallography of a new concept of proteasome inhibition. So we have sophisticated proteasome inhibitors, natural compounds, a lot of them. When we look at the electron density of the wild type, that's the catalytic triad activating the threonine 1-gamma. You see it here on the left side. We also, since we are fast, got interested in determining a rather, from the chemical structure, boring compound, a sulfonyl fluoride, as you see it here on the right side. For us, it was interesting because it is out of frame, because for chemical reasons, you need here a methylene group inside. You are displaced. Now, when you determine the electron density, that is what you get out. And it was, I could hardly convince a PhD student that this is interesting, because this is very interesting. You don't see the compound, but you see that the active site, which is expected to have low temperature factors, which has to be well aligned, is not aligned. So there has been something going interesting. So that means we should get inspiration. We should not look only about that we want to visualize something and describe a picture, at least for talking about the users where I'm belonging to, not talking about developers, but to get this inspiration so that compound for the first point of view does not look that interesting. And when we look at, I can even enhance it here, the methyl group is that. So that is an FO minus FC density contoured at minus 3 sigma. So it does not make any sense at all why a compound removes the methyl group. It is completely nonsense, but that makes it interesting. So the compound does not look interesting. And the system is the following. So what we do is, the first step is we attack the threonine 1-gamma, again with sulfonyl fluoride by release of H plus F minus. And that means we made out of a nucleophile a very good leaving group. And that means since we deprotonated our Bronsted acid, so the threonine 1N terminus, That can now nicely act as a branched base or as a nucleophile, and that is exactly what's will happening. You can perform an SN2 reaction, as it is shown here in red, and you perform an aziridine. And when you place it by an SN2 reaction with an aziridine, it perfectly fits. So that means you need interpretation. Now the next thing is, so that the lysine 33 is completely unhappy. It is protonated, but the 3-1 gamma is absent. And it's in close proximity to the aspartate 17. So aspartate 17, of course, the protein is delocalized. We're having another branched base, which wants to have something. And this is in proximity to the aziridine ring by 3.3 angstroms. So we take the crystals which appear by 6.8 angstroms and place them to 7.5 angstroms. And when you do so, you click it. It reminds me somehow a little bit to GFP or something like that, where you form an imidazole ring on main chain atoms, which gives the nice color of GFP. So we can say that when we make simple chemistry, we can collapse a complete active site, and we can use different bases, the 3-1 N-terminus, the lysine 33, and the 3-1 gamma. So that means it again shows that proteins and enzymes, not proteins, but enzymes are stupid. Because what are they doing? They are catalyzing. They are catalyzing reactions, and you can exploit it. So we're having information and execution. And that explains that you can go in another way, and as I've mentioned it here, new principles, that you can completely collapse active sites by simple compounds, by thinking about chemistry. So I want to continue that what I've talked now, that this was focused more on the proteasome in general. Here I want to continue because the system is even more complex. And that is shown here. So what we have discussed so far is that we're having denatured proteins. They are recognized by the proteasome, by the 20S proteasome. Once it is folded, it is in this way, it needs a ubiquitination, a recognition, and so on. We're having peptides, and I so far was only focusing about the life cycle of proteins. That means you start by individual amino acids, and you end up by individual amino acids. In mammals, it is more complex. In mammals, it is therefore more complex because some of these peptides can escape, and they can be presented on MHC class I molecules to the immune system. And the question is, what is going on? Now, we're having in mammals, and I introduced you to this in the initial slides, various types of proteasomes. We're having, and I only will focus on the so-called constitutive proteasome, cutting complexity down to size. I always use this title because it was a very nice placement in this News and Views article. In going down, but we need to somehow address the immune system as well. And that means what we see, there is only a minor production, which is shown here in red, from the constitutive proteasome, which fits in MHC class I molecules. But the immunoproteasome produces a lot of them. So we need them, so we have a production of antigenic peptides, a presentation to C cells, and the stimulation of the immune proteasome. Of the immune system. So that means it is biologically very important, and we're having now two completely separated pathways. From amino acid to amino acid, from a protein to a presentation by a monological response. I will only focus that is the subunits which are getting replaced, the subunits beta 1C, C for constitutive, beta 1,2, and beta 1,5, these are the three proteolytic active sites. We focus only on beta 5, get replaced by beta 1i, beta 2i, and beta 5i. So that means that these subunits, as I've mentioned previously, that the assembly of the proteasome is very complex. You cannot just take out one active subunit and place it in another. It is a de novo synthesis. So that means when you're having a viral infection, the synthesis of the immunoproteasome takes time. It's the incubation time, and of course it needs degradation. Because when you're having a too high concentration of the immunoproteasome, I will come to this, that's also pretty bad. So what's happening in this? I will focus only on beta 5I, as mentioned, that when you, again wrong, that was very bad. Can you please put it to the picture where I was? Because I have no computer here, to the picture where I was. Otherwise, I go through, oh, it's here, yes, that's fine. So, thanks. So that means once we're having an imbalance and a too high presentation on MHC class 1 molecules, it can well be that this is due to a much too high concentration of the immunoproteasome. And we're having a lot of diseases, as it is shown here, and these diseases are demanding nowadays and definitely in the future, as you can see it here. We have rheumatoid arthritis, diabetes mellitus type 1, multiple sclerosis, and so on. I do not want to mention that the cause of these awful diseases is the proteasome, but it is involved. It can be involved. That is what I want to say. Therefore, as I've mentioned before, that by producing proteasome inhibitors for cancer therapy, we're having other targets as well. Now, we got interested, of course, in therefore how they differ. And that was a great collaboration with Groll from Constance, who is an immunologist. We don't, we are biophysics and chemists. And it is in a way that you're taking a mouse, as it is shown on the left side, and you're making a lymphocytic choriomeningitis virus infection. So that is a viral infection. Then you have to wait about 10 days. Otherwise, the immunoproteasome is not produced in too high concentrations, and the constitutive proteasome is still there. So protein degradation itself occurs by autophagy, and therefore this needs time. And when you produce it in a way like this, you're getting about more than 95% of immunoproteasomes in these livers, and you can purify them, that's our purpose, and to crystallize them and to determine the structure. Now, the importance of the collaboration with Groll was that you need knockout mutants for also analyzing the constitutive proteasome. Our purpose was to visualize and to identify proteasomes from the same species, because that we can directly compare. And here you need this bulb C mice with a beta 2I and beta 5I knockout, because they cannot produce immunoproteasomes. You have to know that in liver, there also exists a content of 10 to 20% of immunoproteasomes. And that will be not helpful. We need homogeneous samples. That is the reason why this was going through this tour de force. Purification, crystallization, and structure determination happened. Now, we have now the crystal structures determined at Bragg-Price resolution of 2.9 angstroms. And that means, why are we interested? We are interested in it that in 2009, Mahamual et al. Published an inhibitor which they screened by combinatorial chemistry. And they figured out, as you see it here, that this is an inhibitor having the epoxy ketone. I introduced it to you. That this is rather specific in the nanomolar range for, this is mouse, this is human, for beta 5I, but tenfold less active against beta 5C. So that's from the constitutive proteasome in both in mouse and in human. Interestingly, yeast is just in between, but this does not only have one type of proteasome, another constitutive one, or an immunoproteasome. So thus, we got interested by determining the crystal structures of the constitutive and the immunoproteasome. The value of this work is because we aimed to compare the APO structures with the ligand-bound structures. That is the value of the work, because when we would only have the APO structure, there would be less explanation. And that is what I want to show you. It is shown here. These are experimental electron densities. You can place it either as a 6-membered ring. It's work from 2012, a 7-membered ring. I mentioned this about, it's unimportant, you place it as a 6-membered ring. And you see well-defined electron density, that is an end cap which slightly removes, but you see that's looking identical. And that also I want to show you here that we are in the area of electron microscopy, but we need high resolution. And that means electron microscopy certainly, and we will hear this in future talk from Henderson, can visualize side chains. But for these purposes, what I want to show you, it is my opinion that there needs further investigation to get such resolution. So I'm a little bit concerned when I'm talking about resolution. I think that the value of resolution somehow gets lost. It's a fight of high resolution, if it is either from protein crystallography or from electron microscopy. So we want to learn something, we want to see something. And there's definitely very nice purposes for electron microscopy, which crystallography can never address, but vice versa as well. That is I want to mention here also. So what I would say is, this is the ARPU structure on the left side of the immunoproteasome. That is from mouse. And keep in mind, we're having two residues, methionine 45 and isoleucine 35. And that is now the structure superposition, or let's say in a kind of movie, because I don't want to show movies, because when we're averaging 10 to the 12th time, I'm not allowed to show movies in this respect. We're seeing a flip. And this causes only minor changes. Ligand fits into subunit beta 5I, slight structural rearrangements. You see it here, I make it again. Of course, you see movement. It's something like a little bit of induced fit. We know this. It's in every enzyme. But we see that's a slight movement of these two residues. Also, when we look at this helix, it's a slight movement. Now, when we compare it to the constitutive proteasome, now look at methionine 45 and isoleucine 35. It's a big movement, you see? It's a big movement. But you need good resolution to see these side chains. And you see when you look at the helix, it's a big movement. It's about a 1 angstrom movement. See this here. Also the neighboring subunit beta 6. So we understand why this epoxy ketone, that it clashes with methionine 45, that it causes major structural reactions. That is the problem. So we're having two completely different pathways, cutting complexity down to size, the constitutive proteasome, presentation to the immune system. Same, identical amino acids. No bioinformatics studies are possible in this. You need experimental data. That is what I want to say here. Now, when we compare it now to the Kyprolis, which is on the market, we see that it does not differ significantly as an epoxy ketone between beta 5I and beta 5C. We know it from structures. But what we can do now is, and this will be done with Overcleft, was a tour de force. It was about 50 compounds which we synthesized, and we go on. We place here D-alanine, the cap, the indane cap is important. This is important. It makes a pi system with a cysteine 48. We're needing a cyclohexyl. We need a lot of input, and that is to the high. I did not went into the fragment evolution approach. The three-dimensional space in chemical possibilities, in chemistry, is unlimited. And the aim is to restrict it by structural research into making an improved development, which is always a refinement principle. So therefore, we did not come up with this compound, but it was a refinement principle to learn out of it. And you see, we significantly can differentiate between beta 5I and beta 5C, compared to this compound. But this is hardly also predictable in my opinion. But computational chemistry is very important, but it always should be combined with experimental chemistry or research. Now, when we structurally compare the superposition of the immunoproteasome and the constitutive proteasome, I was talking about these two residues here, but it is found in all vertebrates that we're having here a glycine 48 and here cysteine 48. And that was interesting. We can also show that there is a helix and they're having a helix dipole, so this is even acting as a nucleophile. I don't want to make this story, however, more simple. So it's not just that you see something, you have to keep in mind the chemistry about dipole activation and so on. And what we then did, and this is exemplarily shown here, and together with Wolfgang Heinemeyer, we performed yeast mutagenesis. We made more than 200 mutants, and our aim was from the structural research to make chimeric yeast proteasomes, because it was so demanding, and in respect to the animals, to succeed in these murine structures. We first of all were interested in the mouse proteasome, because we are not interested in medical doctors for mice, but for humans, so we were interested in the human proteasome. And we aim to understand what's going on. And we could succeed, as it is shown here. We can produce humanized yeast proteasomes. And when we take it out here, so we make now a modeling. And within this modeling, we see this cysteine 48. And we placed here a compound inside and placed in the chloroacetamide and suggested that this interacts with a non-catalytic cysteine. We need a decarboxylation, so we addressed it in a way that we decarboxylate it here, otherwise it will be removed by the 3-in-1-gamma. We know that hydroxyl groups can be hardly removed, but we want to exploit the antiparallel beta sheet formation, and we nicely see. Indeed, it is an immunoproteasome inhibitor. Indeed, we can use structures. Indeed, the structures are powerful. Indeed, we can perform chemistry, as you can see it here. The last topic, for the next or last 5 minutes, I want to talk about proteasome-associated autoinflammatory syndromes. I mentioned to you already that when you're having an imbalance between the constitutive and the immunoproteasome, it's crucial, so you suffer from it. And I took a rather soft slide here, and that means it was diagnosed in patients. And that was done by the work of Kitamura and Arima in Japan. That these poor men or guys having a glycine 128 valine mutation and subunit beta 5I. And when this mutation occurs, that is the outcome. Autoinflammation, lipodystrophy, and periodic fever. Now, since we determined the structures, at least for mice, we got interested in about it. This is the active site I've shown you in the very beginning. Our triads and everything, they're important amino acids. The first triad, I was focusing in more detail, the important triad activating the 3-in-1-gamma. And we see this glycine is beside the serine 129. But this is not involved in an amino acid, which is contributing directly to the active site. So what can we do? So what these guys found is that the proteasome assembly intermediates significantly arise in these patients. We cannot work with these patients, certainly, but we can work with yeast. And when we work with yeast, we can place this mutant, we can place it in beta 2 and beta 5. So this is beta 5I and beta 5. They, however, cause lethality on yeast. So something significantly is going wrong. But we can place it in beta 1, as you see it here. So this is nicely viable. And of course, then we can purify the proteasome out of it and analyze it. Now, when we analyze it, it is in a way that is the wild type. And when you look at this wild type, this is the defined electron density at the active site. That is our catalytic triads shown here. And that is a zoom out. And when we make it now experiment in theory, as it is shown here, we place in the valine 128, we would see that this 3-in-1-gamma would be split, would be shifted. We were very much surprised initially because we can place a beta 1, a beta 5, beta 2, and beta 1 3-in-1 alanine mutation. In yeast, this is survivable. But this mutant only, with the valine 128, only succeeds in beta 1. And now the experiment shows some interesting results. Unpredictable, so you need experimental results. You have here the 3-in-1, and this 3-in-1 is put down. You see, a complete different orientation. Of course, you're having the propeptide, as I've shown it before. I showed you this propeptide of the 3'1-alanine mutation. It completely looks different here. It points down, and you're getting a displacement of isoleucine 126 and asparagine 141. So this whole loop, including serine 129, is displaced. Major structural rearrangements, unpredictable. I keep it short here. We could show by other mutagenesis that this is an assembly intermediate prior to the more complex eukaryotic proteasome. I cite here Hirano. What I've shown you by the proteasome assembly before on structures, that was very simple from a structural point of view. The assembly line of the eukaryotic proteasome is highly sophisticated, highly complex. Now, what we know is that subunit beta 7 is the latest subunit, which participates in the half proteasome. I was showing you archaebacterial proteasomes, archaea proteasomes. Now we are in yeast, and that was the work of these guys. Now, when we look it up, and this is shown here, this is subunit beta 7, beta 1. That was the reason when we introduced the nomenclature from alpha 1 to alpha 7 and beta 1 to beta 7 by the yeast proteasome, that we take the diet, of course, and that was the numbering where we take it out. And you see these are, of course, then from the one ring, and these are from the other ring. Now, when we look at the half proteasome, we see that it is only the C-terminus of beta 7 which protrudes out. It's the latest subunit, and the subunit then eventually attacks the other half ring system, and then maturation occurs. That starts then where autolysis of the prosequences takes place. Now, when we look at the mutant, no, this one is not working anymore. Now, when we look at the mutant, so you see it here, you see this is due to the structural distortion. Now, we can explain why it is in beta 1 stable, because you're having a support of beta 7. But in case of beta 5, which is on the other side, you don't have this support. So you need a contact surface of all of these subunits to place them correctly so that they can mature. And if this is not possible, then it fails. And in case of beta 1, it is possible, because beta 7 supports it. So even if there's no contact here in between, the subunit beta 7 brings these two halves still together, and therefore this was why we can visualize it. So that means for the pure patients, when we have it here, we have a constitutive immunoproteasome, an immunoproteasome, a thymoproteasome, which I've not put it in here, and we see that these pure patients, that they fail to produce immunoproteasomes. And that is the outcome when you produce no immunoproteasomes, as it is shown here. An autoimmune disease, a strong autoinflammation disease, strong disease, because as we can see it in about it, lipodystrophy and periodic fever. But autoinflammation is quite clear why it's happening, as mentioned before. This was my talk. My time is over. So what I wanted to show to you was from structure to application. I decided to talk about the beginning in 1997, 20 years ago, on the yeast proteasome structure. I presented to you assembly, maturation, gating principles. Between 2000 and 2015, we were introducing to natural and synthetic inhibitors. We analyzed mode of actions. The immunoproteasome, we got very excited about it. It's our current topic since 2012. We introduced then from the structural knowledge, these ones, to the proteasome type selective inhibitors. We are currently working with proteasome-associated disease, and this year it's not looking too bad. We are working with the thymoproteasome. I'm grateful to my current team, my technician, Feicht, to Wolfgang Heinemeyer, my senior scientist, and who come up then later from the Wolf Group, who retired, to me and makes the yeast genetics, to my former PhD student and current postdoc. You don't need a lot of people, but you need good people. Hoover, and to my alumni that are my former PhD students. And I want to mention in particular the collaborations. Hoover. It started 22 years ago when I was a diploma student. I published my first structure on the yeast proteasome in 1997, 20 years ago. But the work would not be possible with lots of collaborations. And I listed the PIs and the most important ones here. Thank you for your attention.
So we have time for a few minutes of questions, and then there will be an appreciation from the local organizing committee. So Bodan Schneider.
Yeah, I'm Bodan Schneider from Prague. So if I can ask how proteasome manages not to digest itself, and then maybe it's too specialized, but what is the connection to how interferon gamma actually induces this generation of immunogenic proteasome?
The proteasome, so I did not mention this at all, is in a way that you need a recognition system, and that is ubiquitination. So you're having a very complex system, and via the ubiquitination, you giving a post stamp, a kiss of death, and then these proteins are dead, and they are degraded by the proteasome. Nevertheless, the system is more complex because you have also ubiquitin-like proteins, but it's in this way. The gamma interferon induction, so you're having also MHC class 1 molecules on it, the TAP transporter and so on, but also the immunoproteasome subunits. Beta 1I, beta 2I, which I did not talk about it here, and beta 5I. And that means you're having a permanent production of the proteasome. It's quite abundant in the cell. It's about 1%. And the de novo synthesis, since you produce now more due to the gamma interferon induction, you produce now more immunoproteasomes. And interestingly, that is not understood currently when you're having a lot of immunoproteasomes. It accelerates the production of cytokines, like gamma interferon and other TNF-alpha. And that is the problem for autoinflammation diseases, that when you're having a too high concentration of immunoproteasomes, that it is a feed-forward system. And that is what I've mentioned. There are currently no drugs outside nowadays as specific immunoproteasome inhibitors, but I promise that they will come.
Yes, sir. Please identify yourself.
Actually, I'm from Hyderabad. I'm a graduate student from CCMB. I have a question. I think it is very near. You are inhibiting proteasomes, but proteasomes are also very important for cell survival also. But how the cell survives when you are inhibiting proteasome?
So either you can inhibit it specifically, that addresses the first question, so the immunoproteasome, but when you're having cancer, you're having a high cell proliferation, and that means what you're doing with these inhibitors, they're going in all cells, they are small molecules, but when they are inhibiting all kind of proteasomes, the cancer cell, which needs rapid protein turnover because of the production of malfunction proteins, of mutants, they need to be degraded. So when you inhibit the proteasome in all kind of cells, the normal cell try to rescue us, but the cancer cell will be killed because of its dust, which cannot be removed anymore. This is an idea, and it is my personal point of view that it is the wrong way to block and to try to inhibit the proteasome, that this is a single study only to target this enzyme or this protease, but it might be worth thinking about combinatorial therapy to include also radiation therapy combined with proteasome inhibition, or to combine a cocktail of inhibitors. So therefore, this is also the reason in my mind, in my thoughts, that why dirty drugs for cancer are not so bad. Otherwise, they would not have survived today.
Okay, thank you very much. Yes, one more. Yvonne Jones from Struby in Oxford.
Hello. I'm curious, for the autoimmune disease problem, or the genetic disease, do you think there's going to ever be any potential for stabilizing the proteasome, or specific forms of the proteasome, in the assembly pathway? Because you were saying that the issue is getting correct assembly of it. Do you think there's any potential for a small drug that could, in certain cases, stabilize proteasomes?
So it's a very important question, and I did not mention it in very detail. Only, as I've mentioned, it was a 20 years research topic. So what is done, for example, you're taking beta cells, and you produce in these beta cells, you produce a GP80, so from these cytomegaloviruses. And these cells, they are not retracted, so these mice do not gain diabetes. But when in this way you make an infection, and that means with an LCMV infection, this mice then gain diabetes because you need a trigger system. So some epitopes from individual proteins are not sufficient that you cause autoimmune disease. And what these guys did, it was the Grötrup group, the immunologists, they knocked out this beta-5I. And when they knocked out this beta-5I and make the LCMV induction, these mice, so also they produce GP80, do not produce diabetes. And that was the idea how they screened for PR957, because that was a specific beta-5I inhibitor. That's the rough or short story about in behind. So therefore, as mentioned, autoimmune diseases might rely on immunoproteasomes as well, but they do not need to rely on immunoproteasomes.
Let's thank Michelle again.
Morning. On behalf of the organizers, it's my pleasure to present a token of appreciation to the speaker.
Thank you very much.
Thank you.
Thank you.
Just one more minute, please.
Sorry, I left it in the downstairs. On behalf of the organizers, it's also a really pleasure to present a token of appreciation to Professor Burley for this wonderful session chair. Thank you very much.
Thank you, and on behalf of our keynote speaker and myself, I want to thank the entire audience, and I also want to thank the local organizing committee for organizing a great meeting. Thank you. So it's coffee time, I believe. Right. Thank you.